In this episode, we will talk about how to mimic human metabolism in mice.
For example, neurons, glia.
Let's talk pharmacokinetics for antibody development.
Following the transplantation of hematopoietic cells.
Let's talk how to test if your drug induces a cytokine storm.
Welcome to Jack's Tech Talk with the technical information scientists of the Jackson Laboratory.
So here we are live from SfN. Look at all the people here in sunny San Diego.
We're interviewing a number of our JAX faculty. Also, we're going to bring on board some of our external partners in science who are using JAX mice in their research.
Hello everyone, my name is Neeraj Tiwari and we are here at Society of Neuroscience meeting happening in San Diego, and with me I have Stephen Keegan, who is presenting this poster at SfN.
So we're here talking about some of the work that we've done recently about how we can prioritize targeting Alzheimer's disease hypotheses for drug validation and drug targeting. So what we ended up doing is we took this list, it's called the gene ontology terms, and what that is, it's a functional language to describe what a gene does. So there's a term associated with a particular gene, and it describes this is what that gene, that's the function of the gene. So what we can do is we can take a bunch of these gene ontology terms and then put them into different clusters or groups, or we call biological domains. And these are things that describe disease biology that would lead up to an Alzheimer's disease. Some of these terms, some of these biological domains are going to be things like the immune response or the lipid metabolism or the mitochondrial metabolism. So things that are involved in Alzheimer's disease. And now we just have a really good way to cluster them into very specific things, or specific ways to talk about what's going on in the disease biology. So each of these biological domains are composed of these gene ontology terms. There's very little overlap between one biodomain or one biological domain and another biological domain in terms of the Go terms that make up this biological domain. That's important because when you're talking about these biological domains, you can have a very specific and targeted understanding of the disease biology that's being represented in that biological domain. So when we can take that, we can take the gene list that composes those biological domains, and then that becomes a gene list of interest to us because you need a list of genes to kind of move on with this sort of analysis because you're trying to end up with a drug target. So we take this gene list and then we've developed a thing called the TREAT-AD target risk score, which is a combination of a multi-omics score and a genetic score. And basically we've taken all of these genes that were composing of these biological domains. We've taken a whole bunch of data sources. I think there's 27 genetic data sources. There's multi-omics data, so we're looking at transcriptomics data, proteomics data, and we're looking at all of these data sources, and that's what we use to build into our gene score. So that's kind of what I was showing here at SfN, where we're now providing these tools to the public, where they have the opportunity to use these biological domains to effectively communicate areas of disease biology. And we also have a list of our ranked genes that are associated with Alzheimer's disease.
Hi everybody, I'm here with Sebastian Illes from Germany or Sweden?
Sweden.
And he has done remarkable work with human brain wave-like activity in iPSC 3D cortical circuits. So I wanted to ask about, how is this work relevant? How are you using this platform that you have developed? It's fascinating.
Okay, yeah, great. That's a good question. So I think a key thing for neuroscience is to understand human brain wave function, also dysfunction. For that, one need a model where you can access the full spectrum of human brain cell functionality. And that requires then also to making cells in the way that they can make spike in an oscillatory activity, as you can see from a human brain.
Okay.
And we have developed such a platform where we're now able to assess disease processes and how they influence human brain cell functionality, but we also can assess compound effects of the full spectra. So actually what we see maybe in an EEG of a brain, we can now assess now in a vitro model. And it is scalable, and we are really happy to collaborate with people to advance our platform further.
Okay, fantastic. Congratulations.
Yeah, thank you.
Hey, could you tell me a bit about your lab, and then you go and talk about your postdoc?
I'm the chief of the section on neuroscience at the NIH in the National Institute on Alcohol Abuse and Alcoholism. And so we try to understand how genetic mutations and or environment alter neurocircuit function to alter behavior. So primarily we look at autism mouse models because you have great traction between the human phenotype and the mouse model. So you see a lot of the symptoms that are present in a human replicated in a mouse.
It's really cool because that's one of the things that we really kind of like lose track of, that translatability. How well is that model going to translate to a human condition?
Exactly. So one of the key models that we use is the SCN2A haploinsufficient mouse model. And in that particular mouse model, not all the phenotypes translate over perfectly. However, we do see a lot of them. Like for instance, the presence of absence, epilepsy, seizures, those are present in these mice. Social interaction deficits are present as well. I don't see too much repetitive behavior, but sensory deficits are also present. So just to remind folks out there, like 95%, upwards of 95% of individuals with autism spectrum disorder exhibit some sensory abnormality. So that tracks well.
Awesome. And with this model that you've got now, I noticed that in the poster that you're presenting, there was this like fever phenotype. Yeah, could you talk more about that?
Yeah, so we study the fever effect. So the fever effect is this really mysterious phenomenon that was observed in children as early as the 1950s. And what they found is that certain children and adolescents that experience a fever, they experience improvement in their symptoms shortly before and during the fever. So over the years, obviously, more and more attention was brought to this anecdotal report, but what has since transpired from the 1950s to now is that we have a few studies in humans suggesting that in 4 different studies, either you cannot benefit, or if you do, at minimum 70% will benefit, upwards to 83% of children and adolescents. So what we try to do is try to understand the mechanism mediating the fever effect. Is it solely driven by the immune response, or is it driven by the thermal aspect, or a combination of the two? And what we found is that, you can have the two components, and they can definitely lead to improve autism-like behaviors. Here we focus mostly on cognitive function. So we challenge mice to perform a particular task, locate an escape hole amongst 19 other holes that they can choose from, and there's only one correct choice. So the mice have to be a little bit smart. They have 60 seconds to locate the position of the escape hole. And interestingly enough, we found that the thermal effect was able to decrease the time that they took to find dramatically the position of the escape hole.
And the thermal effect, what you're doing here is you're just warming the mouse up.
We're just warming it, we're exposing them to infrared light, which is equivalent to sunlight, essentially, right? Very concentrated sunlight. And we're carefully monitoring your body temperature. And so what we see is that not only on that particular task, but if we challenge them using a very difficult task where they have to determine whether whether an object is smooth or rough, basically a difference in texture, and associate differences in texture with novelty. It's pretty challenging. I even had one of my students couldn't even do the task. So, even though I could do it, he couldn't do it. So the ones have to do this task is distinguish between the two. And what we found was that the SfN mouse, in the absence of fever, and here I'm mostly referring to the thermal aspect and the immune aspect. So in the absence of the immune aspect, what we see is that just in the thermal aspect, the mice don't have a preference for whether objects that are familiar, objects are novel. They like everything, so they are interacting with both. But that's not normal. What is normal is for them to gravitate towards things that are not familiar, but gravitate to things that are novel to them. But when they're exposed to about 30 minutes or to an hour of infrared, they're able to correctly balance their choices. So now they're going to prefer the novel. So it seems that the effect of the thermal aspect is to decrease their liking, preference for the familiar. So that's the effect we're seeing in sensory circuits.
Cool. That is really great work. I have with me my Jack's colleague, Courtney Hatton. Courtney, you're in Rob Burgess' laboratory. Hey, tell me, what's your project in that laboratory?
So for the past year or so, I've been working on SARM1 inhibition in CMT models of, CMT models.
Cool, fantastic. Look, I know that you've got a poster here at SfN. Hey, tell me a little bit about that poster. Give me that quick 2-minute blurb about it.
So my poster is actually about the SARM1 inhibition. We have 3 different models on there. 2 that was with collaboration with the Cat Lutz Lab, and 1 is actually a model that we're about to publish on the GGB1 CMT1X mice, and we bred those to a SARM1 knockout and evaluated if the SARM1 was helping, the inhibition of SARM1 was helping those models. Spoiler alert, it's not. But I'm working on other models now, so hopefully at a conference near you soon, I'll be able to give more updates on that.
Sweet. Tell me a little bit about the research that you're doing.
Yeah, so I am a 3rd year PhD student in the Tufts Neuroscience at JAX program in Rob Burgess' lab, and I focus on peripheral neuropathies and mitochondrial translation implications of those peripheral neuropathies. So basically I'm studying this protein, mitochondrial translation release factor in rescue, or MTRFR, or C12, or 65. And when it is deficient in the cell, it leads to stalling of mitochondrial translation, and that leads to these peripheral neuropathies and childhood onset diseases.
Awesome, fantastic. And you have a poster here at Neuroscience, correct?
I do. I have a poster that starts in about half an hour. So, yeah.
Fantastic. So, tell me this. You've been here for a while. What are some of the cool things that you've seen here at SfN?
Yeah, so I think my favorite session so far was the mitochondrial energy metabolism homeostasis talk yesterday morning. There were a lot of really great talks about neuronal implications of mitochondrial stuff, which is kind of a niche subject, but it's my niche subject, so I was super happy to find it. Yeah. And there's been a ton of different models, and just all the different types of research is really astounding.
Fantastic.
Hi, my name is Haley Mendoza-Romero, and I am a graduate student at Vanderbilt University. Right, so I'm looking at changes in pups that are exposed to maternal high-fat diet during lactation versus ones that have normal chow diet. Historically, we know that AGRP terminals are decreased from maternal high-fat diet, but there's not really a molecular mechanism that is explained in the literature. So I wanted to see if microglia contribute to that. So I'm using the JAX animal AGRP-Cree crossed with the synaptophysin DD tomato animal to visualize terminals and doing immunohistochemistry for the microglia to look at their interaction.
Awesome. So how much time do you have left in your graduate work, and what's next?
I have about a year left, and soon I'll be applying for jobs, and I am looking forward to exploring all of the possibilities I might have. I would love to continue to work with doing bench work and imaging and microscopy, so those are the types of positions.
Awesome. Thank you so much.
Thank you.
I have Elizabeth here. Hey, Elizabeth.
Hi.
Talk to me. Talk to me about your research, what you're doing. Give me the quick spiel of it.
Yeah, I'd love to. I'm always excited to talk about the stuff that we're doing in the lab. So I work at University of Buffalo, and we're in a motivation and specifically addiction lab. We do a lot of polysubstance use with like nicotine, alcohol, cocaine, heroin, and we're currently also looking into psilocybin work and how that can help with relapse for different drugs.
Fantastic. That's great.
Yeah.
Talk to me. What are you doing? What's your research?
Yeah, so I also work, I'm from University of Buffalo and I work for Dr. Greg Loney. Our lab also looks at polysubstance use, and some of the work that I've done has been on nicotine and alcohol interactions. I am especially studying rodents, so rats, and my current work is on alcohol intake and compulsive like alcohol intake, and we're also interested in like brain regions that are behind some of the perpetuation of alcohol use, and the insular cortex is one of our key areas we study, and so right now I'm looking at different dopaminergic projections from the VT to the insular cortex and how it affects alcohol use.
I am here with Tim Hines from the Jackson Laboratory, and we just got finished presenting in one of the Nano Symposium sections. Tim, why don't you tell us a little bit about your work and what you presented on today?
Sure. So I study a disease called Charcot-Marie-Tooth disease, and it's a rare neuromuscular disease. So it affects the nerves that innervate your muscles that help you move, and those axons or those nerves degenerate in the disease, making it more difficult to move and get fatigue more easily and things like that. And so we have a bunch of different mouse models of the disease. So they have mutations in genes that patients have mutations in. Some of them have the exact same mutations as patients. Some of them have different mutations in the same gene. But they're all pretty useful for studying the disease and how it progresses, especially considering it's a rare disease. You know, we can't really get a lot of like patient tissue samples and things like that. So the mice have been really helpful in that regard.
So you mentioned you have some knockout models and some knock-in models. Can you elaborate a little bit on those different models?
Sure. So part of how the disease progresses for the forms of Charcot-Marie-Tooth disease that we're studying, it involves expression of this other gene called ATF4, which then causes a bunch of stress response genes to be expressed. And we're not really sure what those are doing, so what I'm doing now is knocking out that ATF4 gene in the mice that model the Charcot-Marie-Tooth disease and seeing if it makes the disease better, if it makes it worse, seeing basically what happens.
And I know you said you had a small N number so far, so we have to take the results sort of
yeah, I was hesitant to even show it, but
yeah.
With me today we have George Murray.
My poster at SfN 2020 was centered upon Charcot-Marie-Tooth disease, CMT1A, which is an inherited peripheral neuropathy caused by duplication of peripheral myelin protein 22. It causes weakness and loss of sensation in the distal limbs. So in about 2019, the Inherited Neuropathy Consortium performed a patient-only genome-wide association study. They were able to enroll more than 900 patients, and that's pretty incredible for a rare disease. And they found associations between the strength of foot dorsiflexion, which is decreased in CMT1A, and intronic variants in a gene called CYP1L2, and not very much was known about that gene. Some follow-up in vitro studies performed by the group that published this genome-wide association study showed that by decreasing CYP1L2 expression, you could also decrease the expression level of a key myelination transcription factor, SOX10, and cause a corresponding decrease in the causal gene PMP22, which is duplicated and overexpressed in CMT1A. So that was our mechanism: decrease CYP1L2 to decrease SOX10 to decrease PMP22 and hopefully help. It worked in vitro, and we needed to test it in vivo. We used the C3PMP22 mouse model of CMT1A, which has relevant phenotypes. For example, decreased wire hang duration and decreased nerve conduction velocity, which you also see in nerve conduction velocity decrements in patients. However, when we crossed these with a CYP1L2 knockout, we didn't really see improvement in neuromuscular phenotypes, but we did see an effect on myelination. We looked at femoral motor nerves, specifically the motor branch of the femoral nerve, and we found that CYP1L2 knockout increases myelination. So that's kind of interesting that it increases myelination, but it's not enough to improve nerve conduction velocity, which was a bit disappointing. We did some gene expression analysis, and we found that CYP1L2 knockout remarkably reduces the gene expression pathway disruption signature of CMT1A in this mouse model. However, it didn't affect cholesterol biosynthesis, which is increasingly recognized as one of the primary disruptive processes of CMT1A.
So we're interested in the genetic aspects of developmental exposure to alcohol and what it does with the brain. And we started by looking at cell death, because cell death is a really ubiquitous thing that happens after alcohol exposure. And it can have really deleterious consequences in the developing brain, because not only are you killing the cell that's there, but you may be killing its progenitors, or some of its targets may not get inputs, or what's targeting it may not get inputs. So it can have a lot more severe consequences than what you would have in an adult. So we've done a lot of work looking at different cell deaths. And we used several different models, including one with prenatal exposure and one with early postnatal exposure, to encompass what's called the brain growth spurt, which the third trimester equivalent in humans happens entirely postnatally in mice and rats.
Hi, I'm Jessica. So when I joined the lab with Dr. Kristin Hamre, she had just published a paper looking at differential cell death in the CA1 region of the hippocampus using BXD recombinant inbred strains of mice. And what the BXD mice are is they're a cross between the B6 and a D2, and they're inbred for over 20 generations, and there's now over 200 strains of BXD mice. And she had used a handful of BXD strains and found differential cell death in the hippocampus. And with that, she was able to use genetwork.org in order to map a quantitative trait loci, in which could be point out chromosomal locations possibly mediating the cell death response after postnatal ethanol exposure. So next, within those strains, or within the chromosomal locations, no big genes jumped out, but I wanted to continue the work. So the next step was to do a microarray analysis, in which I tried to make my own normal distribution by picking low cell death strains that showed low cell death after ethanol exposure during postnatal development, and strains that showed high cell death compared to saline controls. And then I also used the BXD, or the parental strains that showed medium cell death. And basically with that, I looked at gene expression changes that were differential across these strains after alcohol exposure. And looking at that, the ones that met my criteria for a high fold change among the strains, and also were significantly different for a strain by treatment interaction, two of the genes were actually within the original QTL. So this proved as almost a proof of concept going back. So one of the genes was transforming growth factor beta 3, which showed differential expression among the strains. And then I went further to look at the suggestive QTL, which also had genes that related to enzymatic activity of TGF beta 3. And then finally, on genetwork.org, I was able to look at my original cell death phenotype and correlate it with the microarray data, and look at genes that were correlated with our original phenotype, and genes that were significant for a strain by treatment interaction that also were correlated with the phenotype included the receptor for our gene, TGF beta 3, as well as other genes that are related to cell death and also neurodevelopmental disorders.
Wow, very cool.
I'm Jessica Biddinger. I'm a postdoc at Dr. Richardson Illes' lab at Vanderbilt University, and I'm working on a project where we're giving mice a high-fat diet exposure during lactation and measuring their anatomical connectivity and neuronal activity response profiles in response to viscerosensory stimuli. And we find that in general, mice exposed to high-fat diet during lactation show blunted satiety responses and blunted neuronal activity profiles. But we're working with Inscopics to get neuronal activity profile readouts from these neurons. And if I play you this video, you can see the neuronal responses and the traces in response to different viscerosensory stimuli. This is an animal that was fasted overnight, and you can see that in response to the refeeding, that the neuron responses in general, that the activity profiles of the neurons are blunted.
Hello, Dr. Burgess. You're Jack's faculty. Tell me, what's going on in your lab? What's your lab focused on?
We're working on a variety of mouse models of different rare neuromuscular diseases. And the predominant one is a disease called Charcot-Marie-Tooth disease, or an inherited peripheral neuropathy where you have muscle wasting and sensory deficits, mostly in your hands and feet.
Cool, that's fantastic. And, Charcot-Marie-Tooth, what are the interesting things about that disease phenotype? And really, are there any good mouse models out there that really highlights this?
Charcot-Marie-Tooth disease is actually a really interesting disease, and one of the reasons I think it's interesting is it's a whole constellation of diseases. There are actually 100 different genes that, when mutated in humans, can cause Charcot-Marie-Tooth disease. So I think what that's telling us is there are 100 different ways that you can get axon degeneration in your peripheral nervous system. So it's really almost like a genetic cell biology study of the peripheral nervous system in humans. And while there are 100 genes, I'd like to think, or I'd like to hope, that maybe it's not going to be 100 unique mechanisms in 100 different diseases, but there's going to be some overlap between those. But there's still going to definitely be dozens of different mechanisms involved in this process.
Fantastic. Have you seen anything today here at SfN or during the time that you've been at SfN that really stands out?
I think one of the things that's really interesting, my lab, about a year ago, we published a couple of papers identifying this cell stress pathway called the integrated stress response as actually contributing to one form of Charcot-Marie-Tooth disease that we're studying, this Charcot-Marie-Tooth type 2D. And when the integrated stress response is activated, the disease actually gets more severe. And that's exciting because there are drugs that inhibit the integrated stress response, and that makes them good targets, good candidates for actually treating this disease, which currently doesn't have any treatments. And the exciting thing here at SfN is that there have been several posters on other neurodegenerative and neurological conditions, even just nerve injury, that also activate the integrated stress response. So I think that'll help, gin up some excitement around developing these therapeutics, not just for these rare diseases, but for a lot of different candidates.
Cool. Talk to me about the research that you're doing here and going on with this mouse model that you have.
Yeah, so this is my favorite story from my lab, the Cox lab. It starts with creating some mice with some really rare diseases, spinal muscular atrophy, respiratory distress type 1, and Charcot-Marie-Tooth disease type 2S. All the way ending with getting FDA approval for investigational new drugs in treating children. So first of all, the Cox lab is really invested in creating mouse models of neuromuscular degenerative diseases, from ALS, Duchenne's muscular dystrophy, to these guys. So the two mouse models I'm talking about today are the EM3, SMARD1-like mouse model, so that's spinal muscular atrophy with respiratory distress, and the EM5 are Charcot-Marie-Tooth disease type 2S. They both involve mutations in this gene called IGH-MBB2. It's a helicase, and we don't know too much about it, but we know that if you have recessive and deleterious mutations in it, it can cause major problems, especially with SMARD1. In humans, it typically causes death before you become 3 years old. It causes severe muscular and neurodegenerative atrophy and respiratory distress, and there's no cure. Kids are usually just put on ventilators and if there's any malfunction of that ventilator or they catch pneumonia, that's it.
Very
And-
severe disease.
Yeah, and it's heartbreaking, especially since I think there's only like 60, 100 diagnosed cases in the world. So when parents get this diagnosis, there's not many people they can go to. And that's what we're here for, to give that hope.
Cool,
And on the less severe end, you can develop less severe motor and sensory neuropathy, the Charcot-Marie-Tooth disease type 2S. You don't typically have that respiratory distress, but it can still cause debilitating effects where you're wheelchair bound and just decrease your quality of life. So we're super thankful that after creating a bunch of these mouse models and talking to patients, our EM5 mouse model is actually based off of a human mutation. The Meyer group at Nationwide Children's Hospital contacted us to try some clinical vectors. And we tried 2 different promoters. I guess to kind of summarize it, we tested these vectors really early on in our mice to see if they'd even work. And with our SMART1 mouse model, they die at 3 weeks. They're super severe. By the time this vector would express itself, they're already showing symptoms. But miraculously, this treatment works super well. These mice that were dying at 3 weeks, they're living past like a year and a half, and they're strong.
Oh, this is really cool.
They're not just breathing, they're thriving. And my favorite picture to kind of display this miraculous difference is these leg muscles. So this is the equivalent of your calf muscle. A non-treated SMART1 mouse, it's super tiny compared to a wild type mouse, not affected. But if we give them either treatment, so we tested a couple of vectors because we want to give patients the best vector. You can see they're like 2 to 3 times the size of our non-treated mice. And even more miraculously, in our milder mice, the ones that start showing phenotypes more around 4 to 6 weeks, either treatment, there's no difference between the wild types. In muscles, in nerves, in neuromuscular junctions, body weights.
This is really cool.
I can't tell the difference. Sometimes they're sometimes they get tubbier than their wild type mates.
So, are you seeing like any neuro-related changes? I know that this is muscle here. Any changes in like neurodegeneration or neural improvement?
Yeah.
That you were talking about?
So, like I said, with the story, we always kind of look at the basic stuff, like are they living past that 3-week time point? And with our severe model, yeah, we were able to get body weights. They're already kind of bigger. And like I said, those muscles look great. But SMART1 and CMT2S are primarily motor neuron diseases. So you definitely have to look at those nerves, right? So that's why we have these pretty pictures of circles. They might not make sense, but this is basically your femoral motor nerve along your leg. And all these little circles are the axons and projections that reach towards your muscles. And once again, we have our untreated mouse, super small. And our wild type, 4 times bigger. And either treatment in our really severe mice, so much better. You can just look at it. Fantastic. But we also see quantitatively, they're awesome too.